All right. Thank you, Bill, for the kind introduction. You kind of stole my surprise. It's a pleasure to be here. Thanks to organizer for the invitation. As Bill said, my lab studies sleep in Drosophila. I believe I might be the only speaker for the 2 days working on something which is not a mammal. So I'll try to keep it simple to a diverse audience. One of the reasons why we work on flies is because I'm really fascinated by the evolutionary conservation of sleep. And I think you should all too as well. One of the questions that we have to still address about sleep is not just because sleep is present in all animals. As far as we know, that's the argument that's been put forward so far. Any animal that has been investigated so far, from jellyfish to whales, show to possess some characteristics of sleep. But the other question that comes together with evolutionary conservation also has to do with the actual evolutionary variability of sleep in these animals. You have animals throughout the kingdom that sleep as little as 4 hours a day. We know elephants do, for instance. On the other hand, you have animals like bats, or most commonly cat, if you have a cat at home, that sleep something like 20, 21 hours a day. And so if you want to understand what sleep does, you have to basically come to argument with this evidence. You have to try and explain why some animals sleep 21 hours a day and other animals can cope with 4 hours a day. Any proposal of sleep function has to address this point. And between elephants and bats, somewhere in the middle, is Drosophila melanogaster. Flies, we know, sleep more or less as much as humans. They obviously are a fantastic genetic model, and their introduction as a sleep model, actually by Chiara's group in year 2000, has really opened a new highway for sleep studies, not just in terms of genetics, but also in terms of physiology and sleep function. And so the question that I try to address today is the following one. Is sleep a vital necessity? So what do I mean by vital necessity? It'll become clear as I talk about the experiments we did. But a vital necessity for me is something that is as a cell biological definition. So food is a vital necessity. If you don't eat, you die. Okay? So is there a component of sleep that is like food? We know a lot about the biology of food. We know the role of calories. We know how sugars and other calories get converted into ATP and how this sustains cell biology. Is there an equivalent at the cell biological level of sleep? So this is a very important question, I think, that still is highly unaddressed. Before I get into the results, I want to introduce how sleep is studied in flies. And this is the system that you might be familiar with. You might have seen this in other talks. It's a system called activity monitor. What it does is, in fact, it monitors activity of the animal. When I started my postdoc, this was the only system basically available to study sleep in flies. It's not the case anymore, but it's still probably the most widely used. And conceptually, it dates back to the 60s, experiments from Simon Benzer. When Simon Benzer basically created the circadian field, he started looking at circadian rhythm in pupae, Drosophila pupae. And what he was doing, he was putting animals into spectrophotometer cuvettes, so that when an animal will emerge from the pupal case, it will somehow interrupt the beam of the spectrophotometer, and the spectrophotometer will record activity on the ticker. And this works conceptually in the very same way. We put flies in a small tube, which is about 6 centimeters in length. There is cotton on one end and food on the other end. And the fly can stay in this environment for days, technically all their life, as long as they have access to fresh food. And as they live in here, all they can do is move back and forth. When they move back and forth, they interrupt an infrared beam, which they cannot see, but stays a laser exactly in the middle of the tube. And by interrupting the beam, they communicate to the computer that the fly is active. If they're walking, they cannot possibly be asleep. Now, this system has been extremely useful for the study of circadian rhythm. As you know, the Nobel Prize for circadian rhythm was basically developed using this system. But it turns out it's not particularly efficient to study sleep. And the reason might be already evident to all of you is that if the flies stay only by one end of the tube, for instance, by the cotton, walking, thinking, dancing, whatever, but not crossing the beam, then no activity will be recorded and sleep might be overestimated. So for this reason, we started working on something that is a bit more, will be a bit more specific. And to cut a long story short, we produced and published actually a couple of years ago this device that we call ethoscope. An ethoscope is a machine that is supposed to be used in a high throughput fashion, so you can still use it to study thousands and thousands of flies a day. It's inexpensive, but it has a critical advantage over the activity monitor, and that is that it's able to use video tracking to look at the flies and see what they're doing at any given point, and any given point in time and space. Most importantly, what we do is not just motion detection, but we actually employ some kinds of basic machine learning to distinguish between different patterns of activity. So we can find when a fly is walking, like in this case, in the tube. We can identify when a fly is actually immobile, properly immobile, wherever they are. But we can also detect whether they are feeding, so performing small movements by the foot, or whether they are, for instance, grooming, so cleaning their wings, which is something that, as you know, flies do very often. And this category is what we call micro-movements, and it's really one of the major new features of this machine, because we're no longer just able to detect when the flies are walking, but we're able to detect also when they are awake outside of the center of the tube. So when you compare sleep as detected with ethoscope to same sleep as detected with activity monitors, this is what the picture you get. So you have on the continuous line sleep recorded with the ethoscope, on the dashed line recorded with the infrared beam split or the activity monitor for both males and females. And this is the pattern over 24 hours. We keep normally animal in 12 hours of light and 12 hours of darkness. And you see that in both cases, you record most of the sleep during the night. We know that flies are crepuscular animals, so they have a pattern of activity that is greater at sunset and sunrise. Males have a siesta, which has already been described before. Females do not. But the most striking difference is that even though the pattern is similar when you compare ethoscope and activity monitor, the amount of sleep is clearly off. And so, for instance, with the activity monitor, you will think that flies sleep something like 80% of the time, while in fact they sleep on average 50% of the time. And female flies have an even striking difference. You would think that they sleep something like 70% of the time, while in fact they sleep less than 25% of the time. And so this is interesting, but not surprising, at least for when we did the experiment. It was already shown by other groups doing video tracking analysis, by ourselves as well, about 8 years ago. When it became really interesting is the sleep amount shows a greater variability when properly annotated. And so this is a group of 485 male flies compared to a group of 881 virgin female flies, for which we recorded sleep across several days. And we plot average sleep amount throughout the day in these graphs. As you can see, in males, sleep amount ranges from a maximum of 60% in some individuals to something like 20% in low sleeping individuals. In females, the difference is even more pronounced. It goes from 40% to something that almost zero. In fact, and this is very surprising, we found animals that sleep at least 3 or 4 minutes a day using this system. And so when I say that no animals has ever been found to appear to be sleepless, I wasn't lying. So we were very excited because this seems to be almost like one of the first evidence. If you look at the activity tracking of these animals, you see that they walk a lot during the day. And then during the night, they still keep walking, but they keep walking without crossing the midline of the tube. And so that explains why lots of this activity is never been detected before. So this is, in fact, our shortest sleeping female. And you see that she walks a lot during the day, but then during the night, she spends a lot of time by the food, performing in green what we call micro-movements. We confirm that the location of micro-movements is really important to explain this phenotype. What we do is we plot where, for instance, the walking activity happens throughout the tube. So if this is the position, length of the tube, you see the walking is evenly distributed both in male and females along the length of the tube. Quiescence, on the other hand, has a strong bias. And you see that quiescence actually happens towards the end of the tube, but not quite at the very margin. Okay, so they have a preferred spot, which is about 10% throughout the length of the tube by the food side. And finally, micro-movements actually happen exactly by the food, especially in females. Micro-movements happen mostly adjacent to the food. And so the reason is that most of those micro-movements are actually food-related behavior. Right, so we done other analysis on this to explain differences between male and females, but I'm not, I don't have the time to show those data. Instead, I'll jump on the second part of the talk, which is the logical consequence of what we just observed. So we found flies that sleep as little as 3-4 minutes a day. So the question is this a peculiarity of only some lucky or unlucky individuals, or is something that potentially every fly can do? Is any fly in principle being able to survive with 3 or 4 minutes of sleep a day? And so the response to this question has to come from studies of sleep deprivation. Sleep deprivation has been, as you know, fundamental for sleep research, but it always comes with a caveat. And so when we started this line of experiments, we wanted to address the caveat first. The main caveat of sleep deprivation is that the system we have and we use to perform sleep deprivation are always difficult to control. In rodents, for instance, some people use a system like this one, where animals are placed on a treadmill, and they have to walk continuously, because otherwise they will get electrocuted. Painful, but not lethal, electric shock at the end of the treadmill. There are other systems, obviously, which are less invasive, but stress is always something that one has to control for. In flies, most groups will use just an orbital shaker. You put the flies on the orbital shaker, and then every minute or so, you will give a good shake so that they will wake up. This system is probably less stressful, we don't know, but it's still mechanically is quite invasive. And most of all, it doesn't discriminate between sleeping flies and wake flies. It just will give a stimulus to everyone. Conceptually, probably the best we come out with is this system, the Discover Water. I assume you're all very familiar with it, so I will not go into greater detail. But the good thing about the Discover Water is that it will deprive of sleep only the animals when they actually fall asleep. And so we took the Discover Water as an inspiration, and we then modified otoscopes to perform some kind of sleep deprivation, and it will be a control as much as it is, at least in the Discover Water system. The machines we created have modules that plug on the bottom, and one of these modules, in fact 2 of these modules, can perform sleep deprivation. Okay, the way it's done is in the simplest form, you have a motor, and the motor is connected with a pulley to the tube, and so basically whenever the fly falls asleep, the motor will rotate and the fly will wake up. And it looks like this. So if you look at the first flies on the left here, you see that she's not moving for a certain amount of time. You can decide how long you want a certain amount of time to be. This is what we call a trigger, and then it should start rotating like this. Okay. Right, so first thing we wanted to check is whether this way of performing what we call dynamic sleep deprivation, that is a stimulus only when the flies actually stop moving, will be efficient in terms of rebound. And so we perform a different kind of sleep deprivation in which what we changed was the trigger interval. Okay, so we went as low as 20 seconds, meaning that if the flies don't move for 20 seconds, you rotate the tube, to as long as 1,000 seconds. So that's more than 15 minutes. If the flies don't move for 15 minutes, you rotate the tube. But anything between, you still let them sleep. And so we did 12 hours of sleep deprivation during the night, following a number of intervals between this range, between 20 seconds and 1,000. And the result is here. You can see the efficiency of sleep deprivation. In a graph that is not here, you will have efficiency sleep deprivation. And obviously, efficiency sleep deprivation correlates with the length of the interval. But what is most interesting is what you observe in terms of sleep rebound. Okay, so after you interrupt the sleep deprivation after 12 hours, you can look at the rebound and quantify rebound. And what you see is that both in males and females, you have a rebound that is only partly linearly correlating with the amount of sleep deprivation that you've caused. So for 20 seconds interval, of course, where the sleep deprivation is more efficient, the rebound is higher. This is rebound sleep over the first 3 hours the following day. But even on 1,000 seconds trigger stimuli, you have a rebound. So keep in mind the flies have not very consolidated sleep as much as humans, obviously. And so in this experiment, you will have only maybe 4 or 5 tube rotation per night, because this is the number of bouts of sleep that are actually longer than 1,000 seconds, 4 or 5 per individual per night. And yet, you actually see a rebound even after such little disturbance. Okay, so having shown that sleep deprivation can lead to rebound in these animals, the next question is, which is the one we opened with, is chronic sleep deprivation going to be lethal? Can we kill the flies by just constantly keeping them awake? And the literature will tell you yes, based on this paper that was published in 2002. In this paper, what they found is that by doing sleep deprivation with finger tapping on 12 animals, they found that after 60 hours of continuous wakefulness, 2 of 12 animals actually died. And so we performed the experiments using the otoscope, and we took male flies, several hundred male flies and female flies, and we started sleep deprivation when they were about 4 days old, and we continued and continued and continued and continued. And by judging from the previous literature, you would expect them to die more or less at this point, while in fact the experiment went on and on until their natural death. And what we found is that we were not able to kill flies by continuous sleep deprivation. This is done with a 20-second trigger, so we will never let the flies be inactive for longer than 20 seconds. In female flies, there was actually a very moderate effect on longevity of about 3-4 days. In male flies, there was nothing that was statistically relevant. We also performed a kind of Randy Gardner experiment in which we kept flies awake for 10 days to study how sleep pressure builds up in this animal. And so this is the experiment: male flies, female flies, 10 days, interrupt the sleep deprivation after about 10 days, and study what happens after and during the sleep deprivation. And what we found is that after 10 days of sleep deprivation, the rebound that we observed was really nothing major compared to the 12 hours of sleep deprivation rebound that we've characterized before. It was pretty much the same strength and same magnitude. More interestingly, we found that during those 10 days of sleep deprivation, the natural activity of the animals was not necessarily correlating with building up of sleep pressure that you would expect after 10 days, but it was actually correlating with the circadian activity of the animals. In other words, at crepuscular times, when the animal is supposed to be active, it is active. It doesn't matter that it's been sleep deprived for 10 days in a row at 20 seconds interval. At crepuscular time, you need no stimulus to keep the animal awake. The circadian rhythm will still overwhelmingly do that job for you. And we confirmed that, and this is my last experiment, by doing the same experiment in clock mutants, both in a regular LD condition or in DD condition. And what you see is that when you look at the number of stimuli that we have to give to those animals throughout the 10 days, in complete absence of circadian rhythms, this number of stimuli becomes flat, meaning that, again, the main stimulus of activity in these animals is not building up of sleep pressure, but circadian rhythms. Okay, I'm running a little out of time, so I want you to go home with a take-home message. So sleep deprivation is not lethal in flies, whether we induce it or whether we find it in wild-type population. This led us to a working model that we're now pursuing in the lab. And the working model is that sleep is not just one thing. It's not an unicum, not an unum, but it's actually a mix of 3 components. And we like to define a vital component, which is the one that might really work like food. We still don't know how much of the sleep we get or flies get belong to this component, but we still cannot exclude it. But we definitely know that it must be very, very small. A useful component is the one that actually helps your memory, helps your performance, keeps you alive if you're driving a car after sleep deprivation. And finally, an accessory component whose role is actually just to maintain a species-specific sleep amount. And so basically, going back to the original slide on the difference between sleep in bats and sleep in elephant, we propose that the main difference between those 4 hours and 21 hours between bats and elephant actually nails down to the accessory component. And so if it's easy for us to accept that bats sleep 21 hours just to maintain some kind of species-specific rest condition, why can't we accept that the same applies to all other animals? And I'd like just to point out the authors of this, most of the work. So Quentin, who's now in Canada for his postdoc, just graduated in September, and Esteban, who is in the audience, and the rest of the lab. Thanks.
Okay. Thanks, Giorgio. That was a sensational result. So it should be controversial. We're making the young people in the session work extra fast. They have to speak faster. So we've got time for a couple of questions, I think. I can't see, I'm blinded.
Hi, Ivana.
Vlad.
Giorgio, you mentioned 21 hours of sleep in beds. Are you sure those data are strong enough? So what are those original studies that show this?
Can you keep it brief?
Yeah, I mean, there's, one can always argue about the original studies, but there is enough evidence also from other animals, not necessarily in bat. There is evidence, as you know, in cats. There is evidence in koalas. There is evidence that sleep changes a lot with seasonality, and some animals sleep more during summer time of the day, time of the year than others. So I think there is very strong evidence that you know better than me that sleep amount changes a lot between species. Whether you like to call it 19 or 20, the point is still made.
Carl. Then we'll after this, one more question. Okay,
intriguing, but there is duration and there is intensity.
Yes.
So how do you measure arousal threshold in your animals?
Yeah, so basically what we do is we measure the number of stimuli as a proxy for arousal intensity, arousal threshold, because if you can imagine that if an animal has a low arousal threshold, then just one stimulus will be enough to wake them up. But if they have a stronger arousal threshold, higher arousal threshold, then you need 2 consecutive, for instance, tube rotation to wake them up. So the number of tube rotation correlates with arousal threshold.
Okay, one last question, Ivana.
Thank you very much. And I apologize, the clinician, I'll have to ask a silly question. And that is, how do you know that they're really awake even when they're moving? There is obviously sleep and there isn't such a thing as microsleep. So we don't really have intracortical electrodes everywhere. And also,
Well, I can argue that even if you had intraelectral electrodes, you will still be asking the same question, right? Because those are just correlates, same as activity. Activities are correlated very much with electrophysiology. Yes, I mean, it's technically possible that they sleep and walk. But again, I could make the point about any animal model or even human model we study. So
I'll give you a brief
I think it's very unlikely that that's the condition. I mean, especially, for instance, when at circadian times when they're mostly active, they just do what you expect them to do, so feed and walk around and groom. So, they will really have to be zombies to be able to do that.
Okay, thank you very much. Thank you. Okay.